Didactic Residency Edition. I am Dr. O'Leary, a fourth-year psychiatry resident in the U.S. National Capital Region. This is a podcast about psychiatry and neuroscience, and I produce this all by my lonesome because, well, I love reading about neuroscience and this helps me to learn it better. Plus, it's like having dictated notes. If I need a refresher in the car, I can just listen to myself ramble about a subject. Aside from that being a little narcissistic and weird, it helps to remind me how I approached a topic previously. I need to warn you that I am not trying to express anyone else's opinion here, and this includes the Department of Defense, the Defense Health Agency, the U.S. Army, or the Multidisciplinary Association for Psychedelic Studies. When I first started researching this episode, I thought I would try to do a comprehensive review of all the various functions of serotonin across its 15 or more different receptor types, but I soon found myself really overwhelmed. And more importantly, I found that really some stories are just more interesting to tell than others. So I'm going to talk about serotonin and focus on how a few serotonin receptors can not only help us to survive and motivate us to reach our goals, but sometimes convince us that we are fusing bodies with a tree. Serotonin is a neurotransmitter, or more accurately, a neuromodulator. It's generally released in a mode called volume transmission. Volume transmission basically occurs by releasing a substance and letting it reach its receptors by diffusing or just hitching a ride on flowing extracellular fluid like the CSF. They call that convection. This type of transmission is opposed to wired transmission where a synapse is relatively isolated from everything else, and the path that the neurotransmitter, once it's released, can take is more tightly controlled. In general, volume transmission happens where barriers to flow are not quite as tight, and it requires larger relative amounts of the neurotransmitter, in this case serotonin. Volume transmission has the benefit of kind of being more fine-tunable based on what kind of signal you want it to be sent. You can send out larger amounts or not quite as large amounts. You can send things out in bursts or kind of continuously. The most common medications that psychiatrists use are SSRIs, the selective serotonin reuptake inhibitors. These drugs reduce serotonin reuptake in a synapse after it's been released by blocking the presynaptic serotonin transporter. However, it doesn't easily cross an intact blood-brain barrier unless it's carried by something else, and I'll get to that in a second. This is a good thing because the location of serotonin, even though it uses volume control, is pretty tightly regulated. There are these self-proclaimed health experts on social media that really like to point out that most of your serotonin is made in the gut, about 90% of it, and that this facilitates our brain-gut connection. But given that serotonin in the gut does not readily cross the blood-brain barrier and would likely wreak havoc if it did, this particular fact is not actually good evidence for this gut-brain connection. There are other facts that would better explain how our gut and brain influence each other, but that's really not the subject of this episode. The fact that most serotonin is produced in our GI tract is impressive, but not very informative by itself. Some of the most common side effects of our SSRIs are GI upset, nausea, and diarrhea, and this is likely due to the large influence of serotonin on the gut. Although it is probably not making it from the gut to the brain in very large amounts, serotonin that is made in the gut can get to other areas of the body, including the brain in small amounts by hitching a ride inside of thrombocytes, aka platelets, because platelets have the serotonin transporter and can kind of suck up free-roaming serotonin. They do this because serotonin is involved in platelet aggregation and can also cause vasoconstriction, both of which help to stop bleeding. That's why serotonin reuptake inhibitors come with a small risk of increased bleeding. Serotonin also has ionotropic and chronotropic effects on the heart, which means it can affect the strength and speed of contractions. This has caused some speculation that thrombotic events in the heart increase the chance of arrhythmias, not only because of the hypoxia and the resulting cell death, but also because of excess serotonin being released by platelets in the plug, and this has effects on cardiac myocytes. I've told you a lot about what serotonin kind of does, but now I'm going to back up about 1 billion years, because serotonin is considered an ancient molecule. And we think this because it's involved in so many bodily processes and also shared across many distantly related species. It was thought to have evolved first somewhere between 750 million and 1 billion years ago, and possibly evolved as an antioxidant because, well, the Earth was increasing its amount of oxygen, and being an antioxidant would be very helpful. Serotonin is derived from tryptophan, and there are a lot of derivatives of tryptophan that can act as antioxidants. Melatonin, which is a derivative of serotonin itself, has strong antioxidant effects. And melatonin is also widely present across life forms, including plants and fungi. Another antioxidant derivative of tryptophan is psilocybin, which is a fungal compound that we can ingest. And in our bodies, psilocybin is converted into something called psilocin, which itself is a strong agonist of the serotonin type 2A receptors. And this causes, well, among other things, some hallucinations, illusions, a loss of sense of time, or a sense of self as a discrete object. But that's getting ahead of myself. The very first serotonin receptor to have evolved was the 5-HT1A receptor. 5-HT just stands for 5-hydroxytryptamine, which is another name for serotonin. There is a large family of 5-HT receptors, and I'm going to discuss just a few of them today that have relevance to psychiatry. Serotonin itself has been described as something to the effect of being somehow involved in almost everything our body does, but being primarily responsible for none of it. It's like a jack of all trades, but a master of none. Serotonin is a modulator of cell activity, but we best know it as a modulator of brain activity. So let's dive into a select few serotonin receptors, where you can find them, and what they appear to do for us. All serotonin receptors are G-protein coupled receptors, except for the 5-HT3 receptor, which is a ligand-gated cation channel, which means that it lets positively charged ions like potassium, sodium, and calcium flow through it. Remember that G-protein coupled receptors send signals that activate or shut down various metabolic pathways within a cell, and they can also result in DNA transcription. Ion channels are more fast-acting, and they'll either result in depolarization or hyperpolarization of a neuron, which will cause the neuron to fire or to reduce its chance of firing. It seems weird to start with 5-HT3 instead of 5-HT1, but because it's so unique, I feel like it's really a good place to start. So 5-HT3 is probably most relevant to physicians in general because of its location in the GI tract and the area postrema of the brainstem, where it is implicated in nausea and vomiting. Ondansetron, aka Zofran in the US, is a medication that blocks 5-HT3. So this reduces the response rate of its neuron, and in the end somehow effectively reduces nausea and vomiting, especially in patients receiving chemotherapy. 5-HT3, though, is also located on the hippocampus and in low concentrations in the ventral tegmental area, where it helps regulate dopamine release. And so antagonists of 5-HT3 have been speculated to potentially have antipsychotic properties, but we don't have any on the market for that reason. There are, though, antipsychotics that also antagonize 5-HT3, among other things, and these include clozapine, quetiapine, and olanzapine. There are also antidepressants that antagonize 5-HT3, such as mirtazapine and vortioxetine. The cannabinoids, CBD, cannabidiol, and the delta 9 THC, also exert at least some of their anti-nausea effects by antagonizing 5-HT3. 5-HT3 may also be critical for neurogenesis in the hippocampus. So remember that in our central nervous system, there are very few places where neurons regenerate once we're adults. One of the actions of SSRIs when taken chronically is to increase neurogenesis in the hippocampus, and 5-HT3 may play a role in this. Let me give you a little background here. Of those few places in the CNS where neurons are known to continue to regenerate into adulthood, the subgranular zone of the dentate gyrus in the hippocampus is one of these areas. Many chronic mental illnesses, especially like depressive disorders, are associated with a loss of volume of the hippocampus and reduced neurogenesis. This is one of the ways that depression can increase our chance of dementia. In a recent study using adult rats, because, well, their brains are dissectable, and we can't really dissect human brains a lot, showed that giving fluoxetine chronically increased neurogenesis and progenitor cell concentrations in that dentate gyrus. So the cells that make other cells and also new cells increased in number. So they suspected that this was because of the fluoxetine, because it was blocking the reuptake of serotonin, which was making more serotonin available to stimulate the 5-HT3 receptors. And they thought this was what was responsible for the neurogenesis. So to test this, they gave ondansetron, Zofran, which antagonizes 5-HT3 along with the fluoxetine. And this effectively blocked the increase in neurogenesis. I could say a lot more about 5-HT3, especially because it's also present in the spinal cord and in peripheral nerves like the vagus nerve, and it can also help us regulate autonomic activity. But I need to move on. So as a quick review, 5-HT3 is a cation channel, not a G-protein coupled receptor, that when activated increases the firing rate of its neurons. We know 5-HT3 best as a target of antiemetic drugs like ondansetron, but it's pretty widespread in the body. With regard to mental illness, one of the most profound effects it has may be to increase neurogenesis in the hippocampus, a process that is often impaired in chronic mental illnesses. Now let's move on to those G-protein coupled receptors that make up the vast majority of serotonin receptors. Now I can get back to those 5-HT1 receptors. They are very similar to the 5-HT5 receptors, and also similar to D2 or dopamine 2 receptors. Agonizing a 5-HT1A receptor, for example, will reduce the production of cyclic AMP, and can also result in the opening of potassium ion channels, and this will hyperpolarize the neuron. This is similar to what happens when D2 receptors are activated. The end result of 5-HT1 and 5 receptors being activated is really the opposite of that of what happens when 5-HT3 receptors were activated. 5-HT1 and 5 generally reduce the rate of neurotransmission in either or both the postsynaptic or presynaptic cell. They put a brake on neurotransmission, and this can be extremely important. The 5-HT1A receptor is ancient, and it's found in many, many parts of the body. In the central nervous system, the 5-HT1 receptor is in many places, and it's often a postsynaptic receptor in the cortex, but it's especially dense and important in places like the amygdala and the hippocampus and the medial part of the temporal lobe and the hypothalamus. Given that distribution, the nerds in the audience may have already predicted that this receptor has been implicated in everything from regulation of our basic functions, such as like our thermoregulation, autonomic regulation, feeding behaviors, our cognition, our long-term memory, but also complex behaviors, including reward seeking, as well as the regulation of our mood, especially our anxiety states and threat response awareness. 5-HT1A also plays a large role in regulating the release of serotonin itself in feedback to the raphe nuclei. where serotonin in the central nervous system is primarily produced. It does this primarily through actions on presynaptic autoreceptors that tone down serotonin releasing neurons after serotonin has been released. Many papers that I read highlighted 5-HT1A's role in regulating impulsive and aggressive behaviors. It appears that agonists of 5-HT1A, including serotonin itself, can reduce impulsivity and aggression. It helps us to think before we act. As an oversimplification, one could describe 5-HT1A receptor as densely limbic in its distribution, extremely important for primary survival behaviors, but also for adaptive behaviors and regulating our emotional states. The rest of the 5-HT1 receptors, which are like B through F, are not inconsequential, but to keep this podcast less than an hour long, I'm going to kind of skip over them, but I will be coming back to 5-HT1A. I move on to what is currently my favorite serotonin receptor, 5-HT2A. 5-HT2A is more widespread in the central nervous system than 5-HT1A, especially in our higher functioning areas of our cortex. To make it easier to describe the distribution, it may be better to start with places in the brain where 5-HT2A is less abundant. So it's nearly absent in most of the brainstem, and it's very rare in the cerebellum. 5-HT2A is especially famous for its expression on the dendrites of pyramidal cells in layer 5 of the cortex, where it affects all of our higher thinking. You could think of 5-HT2A as helping to govern our advanced evolutionary gifts, such as our executive functions, our working memory, goal setting, our ability to have deep thoughts, our social awareness, how we interpret sensory stimuli and associate senses with memories, or how we associate memories with other memories, or how we have awareness of the significance of the context that we are in, and even how we build our sense of self. Now that was a long run-on sentence with lots of things. 5-HT2A receptors are also frequently located intracellularly, which means that serotonin in the synapse is not stimulating these receptors. These intracellular receptors are on endosomes, and they could be interacting with serotonin that somehow makes its way into the cell. Now, I don't know exactly why this is important in general, but I know one reason why it's important. Many of the psychedelic drugs that hit the 5-HT2A receptor are lipophilic, which means they can cross the blood-brain barrier pretty well, but also easily get into cells. So they could be exerting their effects primarily via intracellular signaling, but that's really a hard thing to observe. So if I were to talk about 5-HT1A and 2A and overgeneralize, I could say that while 5-HT1A helps ensure our survival, 5-HT2A helps to make us uniquely human and promotes kind of flexibility in our thinking. Also, I have failed to mention that while 5-HT1A is generally inhibitory because it hyperpolarizes a neuron, and reducing cyclic AMP and opening potassium channels, 5-HT2A, when activated, is excitatory because it results in closing of potassium channels, less hyperpolarization, and an increase in the firing rate. You may remember from my last few episodes that many antipsychotics block 5-HT2A receptors, and this is thought to be one of their mechanisms of action. The antipsychotic pimavanserin likely acts primarily as an inverse agonist or antagonist at 5-HT2A. While most antipsychotics still antagonize the dopamine D2 receptors, the stronger D2 is blocked, the more likely someone is to develop movement disorders. So that's why pimavanserin is given in Parkinson's disease, because dopamine is already very low, so messing with the dopamine receptors puts the patient at a very high risk of the movement side effects. One of the most studied functions of 5-HT2A is as a modulator of pyramidal neurons in the cortex, especially in layer 5. There, in layer 5 of the cortex, 5-HT2A receptors are densely located on the apical dendrites of pyramidal neurons. But that's not the only place that they are. There are many serotonin receptors in the cortex, but I'm going to focus now on 5-HT1A and 2A and how they interact a little bit. In the cortex, there are neurons that release glutamate. For example, they may extend into the dorsal striatum and release glutamate into the direct pathway. I've talked about that in the past. But they might also go to the thalamus and release their glutamate there. Or they might go and feed back down into some of these brainstem nuclei and release their glutamate there. These glutamatergic neurons send projections all over the brain in order to regulate things and help make associations, coordinate responses. When serotonin is released into the cortex, it will stimulate both 5-HT1A and 5-HT2A, but also 5-HT2C, 4, 6, and 7, which do similar things, but I'm ignoring them for now. So 5-HT1A and its friends, like 5-HT5, have opposite effects from 5-HT2A and its friends, 2C, 4, 6, and 7. But 5-HT1A in particular has a 2 to 6-fold higher affinity for serotonin. Therefore, the location and the density of each receptor relative to where the serotonin is released can determine how it affects the target cell. High volume transmission, where there's like a lot of serotonin released, overall will probably favor 5-HT2A transmission. But low volume transmission will probably favor 1A because 1A has a much higher affinity. Around the pyramidal glutamatergic neurons are populations of GABAergic interneurons that also have 5-HT1A and 2A receptors, and they indirectly regulate the release of glutamate by affecting the pyramidal neurons. So here's a little rundown. Dumping some serotonin into the cortex can result in 5-HT2A activation, which directly increases glutamate release from pyramidal neurons. But it also results in 5-HT1A action, which will decrease glutamate release. And 5-HT1A autoreceptors will decrease the amount of serotonin being released overall. In the same neighborhood, there are other serotonin-responsive cells. Stimulating 5-HT2A receptors on GABAergic interneurons will suppress glutamate release from the pyramidal neurons. But stimulating 5-HT1A on these same GABA-releasing neurons can reduce the release of GABA and disinhibit the glutamate release from the pyramidal neurons. Also, some of these GABA interneurons have 5-HT3 receptors, which are those receptors that are ion channels, and they can increase the excitability. But 5-HT3 has a lower affinity for serotonin than both 5-HT1A or 2A. So it only really comes into play when either there are larger amounts of serotonin being released, or it's kind of the only lonely receptor there. To get slightly more complicated, there are two primary populations of GABA interneurons called the parvalbumin neurons and the non-parvalbumin neurons, and that's due to how they looked when they were stained with a dye that hit parvalbumin. It probably would have been better to name them fast-spiking and not so fast-spiking neurons instead, because this is more descriptive of their function. The parvalbumin GABA interneurons can fire really quickly and repeatedly, and in doing this they tend to regulate the frequency that the glutamatergic neuron fires. The non-parvalbumin GABA interneurons, or those not so fast-firing neurons, fire less frequently, so they tend to regulate more the volume of the glutamate that is delivered. So both the frequency and the magnitude of transmission of a neuron can be regulated in different ways. And this regulation sends different types of encoded information to the receiving neuron, which itself has little receptor antennae on them that are tuned to detect these variations because they have various affinities for these neurotransmitters. Maybe now it's easier to understand why we have not been able to find single genes that cause depression or schizophrenia or anxiety or OCD. But these diseases still have pretty high heritability. Anything that disrupts this kind of elegant balance of this complex system can result in poorly regulated or deranged cognitive functions or deranged emotional states. It can also help to explain why things like environmental factors are so important for psychiatric illnesses. By changing our environment, like having different sensory inputs or being in a different emotional state or ingesting certain substances or even just exercising more than we previously did, the functioning of various parts of this whole system can be affected pretty profoundly, either in the short term or in the long term. To end today, I want to talk about how humans have taken advantage of a disruption in our serotonergic system to basically change the way that they think. Compounds like psilocybin that occur naturally and more modern synthetic compounds like LSD, when ingested, can wreak havoc, a special kind of havoc on our brains. It's a kind of havoc that many people want. They seek it out. Our brain's main job is to predict reality. It needs to tell us the value of our sensory information and very basically kind of whether to move toward or away from something or whether something is inconsequential. We are also able to brood, to kind of think, to create internal experiences, to pull up prior mental states kind of in the form of memories, and at times to spontaneously create novel mental states, some of which are highly valuable to us. We call these novel states good ideas. But good ideas are not always as good as they seemed at the time. Our cortex takes sensory information and our own thoughts, and it associates it with all the other things that are going on in the brain. If the only red thing that you've really ever seen in your life was a cardinal, then the first time you saw a red scarf around someone's neck, you would immediately think that they were wearing a cardinal. And by that I mean the bird, not the Catholic cardinal. When your expectation is violated, an error signal is processed in the brain, and your brain should update its predictions about what, for example, red means. However, we can have biased cognitions that sort of resist being updated. In depressive or anxious states, we often brood on negative thoughts or potential threats, and this gives biased salience toward sensory information that appears to validate those thoughts. For example, she frowned at me, she must be able to tell that I'm a terrible person, or he's walking toward me, he's going to attack. Psychedelic substances like psilocin and LSD temporarily mess with our ability to interpret our thoughts and our senses, and it really causes us to violate our normal assumptions about reality. They do this primarily by hyperactivating 5-HT2A receptors in the cortex, which results in a flood of kind of odd or improbable thoughts, strange sensations, unlikely associations in our brain. Some people even describe something like ego dissolution, where they kind of lose their sense of self. With 5-HT2A agonists, there are especially frequent distortions of visual perceptions. This is why people like to drop acid and then watch a laser light show. There can be just frank hallucinations as well, but often people describe things like hyperesthesia, when senses feel enhanced, like colors just seem brighter or sounds are sharper. They also may describe synesthesia, when kind of one sense let's say the sound of water, results in experiencing a different sensation simultaneously, such as the taste of pears. Often people will say that colors have sounds. People experience mystical sensations and often say that they have deep, meaningful insights or feel like extraordinarily connected to others or to the world around them. They may even kind of lose a sense of where their own body ends and their chair begins. These spiritual insights, if written down and kind of reread when someone is sober, often appear to really make no sense at all. Like, for example, if the chair were to promulgate its effective self, the universe would give pause. Or they could just be vague, meaningless aphorisms like, dude, love is the answer. Not everyone has such rosy, connected experiences, though. Horrendous, frightening experiences have also been described. Someone might describe like having died over and over and over again, or being stolen from their body. But in the end, there does seem to be a strong bias for positive experiences when using these types of psychedelic substances. Even though it's unlikely that anyone is going to figure out the answer to world peace while tripping or the use of psychedelic substances can have enduring positive effects for individuals. It seems that somehow the massive activation of 5-HT2A has enduring effects on the brain. I'm not going to get into too much detail here, but most of the proposed mechanisms on how these substances can treat things like PTSD or depression is by promoting neuroplasticity or flexibility of thought. And this is evident in neurogenesis and arborization of neurons, which basically means that new neurons are formed and old ones make new connections. Psychedelics have a way of unsticking our thoughts from very sticky places, and they do this by massively and selectively activating 5-HT2A serotonin receptors. Thank you for listening. I hope everyone has a great new year. Until next time, I am Dr. O, and this has been an episode of Psydactic Residency Edition.